These mice are born out of two male mice. And this is huge news, but not for the reasons you think. This is gonna be kind of a long one, so stick with me because it's utterly fascinating. You might be surprised to hear this, but we've been actually trying to create mammals using two maternal pieces of DNA or two paternal pieces of DNA since the 1980s. The reason why is because we have a special type of genes called imprinting genes, and they play a very fascinating role in health and disease. Let me give you a very simple example. You see, your genetics is a little bit like a recipe book. You get a recipe book from your mom and you get a recipe book from your dad, and they are for the exact same recipe. Let's just say spanakopita because I'm Greek. But every family makes the same recipe a little bit different, so your dad's copy might say loads of feta and make it crumbly, and your mom's copy might say make it soggier and add more spinach. I don't know. Imprinting genes are essentially very fussy little chefs, and they want to follow just one of those recipes. It will either be maternal or paternal. They will not want to follow both. And a lot of those genes are very important genes, like things that encode for insulin growth factors, hormones that are involved for your development and healthy maturation. The other copy that they don't want to use will be silenced used epigenetic factors, which are things that sit on top of your DNA and are acting a little bit like a maestro that says stop or go. On the way to making bipaternal mice, Lee and his colleagues actually discovered some of the most important things we have ever discovered about imprinting genes. They did this a few different ways, but essentially they used a fertilized egg, removed its nucleus, and then substituted that with the nucleus of another sperm cell. What they noticed in the beginning is that most of those embryos would not even survive a few hours, which is totally expected. But then using a powerful tool known as CRISPR, which is a little bit like a DNA sewing machine, which can find very specific locations and add or take away anything you want it to. And they found some pretty fascinating things, like suckling ability was heavily affected when they knocked out certain genes and was restored when they switched it back on. Most importantly, they found a lot of genetic factors that influence the function of the placenta, a fetal organ that keeps a baby alive while it's still in utero. But the reason why this research is even more important is actually because of stem cells. You might be surprised to hear this, but we can actually take the cells from the top of your skin and turn them back into an embryonic stem cell, which can be used to make any type of cell in your entire body. Scientists have been working on this for over 20 years, but sadly, once cells go back in time and can become anything they want, a lot of problems arise. And a big part of the reason why is because of imprinting genes. To go back to the baking example, our genetics are a little bit like this. We all get more or less the same recipe book. Some of us have a few pages missing, or our copies are a little bit banged up. But for the most part, they function really, really well. And we realized that it's not just about the recipe book we're given, it's about the type of oven we have or the raw materials we use, and so much more that can influence the final result.